Good day, dear students. Here's the second lecture on the course of pathological anatomy, and the topic of this lecture is necrosis and apoptosis. As you know from previous lecture, any cell injury leads to morphologic changes in this cell. These changes, being first reversible, turn to become irreversible and finally lead to cell death. The cell death could be of two types, necrosis and apoptosis. They differ by their pathogenic mechanisms, morphologic changes, and the role they play in normal functions and pathological processes in the organism. The apoptosis could be found both in normal functioning and under pathological processes, whereas necrosis is always a pathological process. What changes could be found in cell under necrosis? All the changes found in necrotizing cell are divided or are grouped into four consequent stages. These stages are paranecrosis, necrobiosis, cell death, and autolysis. The pathologic injury of the cell leads to reversible changes in the cell. These reversible changes are presented by degeneration processes in the cell. Progressing of these changes leads to irreversible changes in the cell when the catabolic reactions in the cell predominate over the anabolic reactions. This happens at necrobiosis stage. The third stage is distinguished called cell death. This is the moment when the cell dies, and morphologically it is almost impossible to identify this particular time. The next stage, usually found and investigated by morphologists, is autolysis. Under autolysis, the hydrolytic fusion of dead substrate of the cell takes place. This occurs under influence of proteolytic or hydrolytic proteins of the cell. In the pathogenesis of necrosis, we can distinguish a few key points. The main thing is ATP deficiency. This leads to loss of selective permeability of cell membrane. This results in cellular ions homeostasis imbalance or impairment. In particular, calcium levels increase in the cells. This induces proteins to become active, in particular proteolytic enzymes which destroy the intracellular organelles. Increased amount of active forms of oxygen and ubiquitin protein also promotes destruction of organelles and fusion of the proteins. Under microscopic investigation, the following changes could be found in necrotized cell. These changes are found in nucleus, cytoplasm of the cell, and also in the intercellular substance. The changes found in the nucleus of the cell are karyopyknosis, karyorrhexis, and karyolysis. Karyopyknosis is shrinking of the nucleus. The nucleus becomes small, the chromatin is condensed, it occurs, and the nucleus becomes hyperchromic. Under karyorrhexis, the nucleus undergoes destruction, leading to such appearance of a number of small fragments of chromatin. At karyolysis stage, the fusion of the fragments of nucleus takes place. Almost same processes happen in the cytoplasm. The pathologic changes in cytoplasm start from protein denaturation and coagulation. This is called plasma coagulation. At plasma coagulation stage, we can find bright eosinophilic cytoplasm presented by coagulated proteins. Sometimes the coagulation process is turned into colliquation. The next stage is plasmarexis, when the cytoplasm is separated into a few coagulated fragments. And the last stage is plasmolysis, that occurs under influence of lysosomal enzymes. What changes occur in intercellular substance? They are fusion of the matrix, that is, fusion of intercellular space, collagenylysis and elastolysis, which are fusion of collagen and elastic fibers. These processes lead to homogenization of intercellular substance, microscopically presented usually by light eosinophilic homogeneous masses. This dead substrate of tissue is called tissue debris. Usually the necrotic area, in particular tissue debris, is surrounded by demarcation inflammation. It is a reactive inflammation around necrotic area. Sometimes we also call it demarcation line. The macroscopic findings at necrosis are also typical. In particular, we can find changes in organ consistency, color, odor, and size. The organ could undergo mummification or malacia. The mummification is the macroscopic finding when the necrotic area becomes dry and dense. Sometimes the necrotized tissue becomes softened. We say softening of necrotic tissue. This is called malacia. In majority cases, the color of necrotized tissue becomes pale, whitish yellowish, as we can see on this slide. Sometimes when the necrotized area is filled with blood, the color of necrosis turns to become reddish. We also say hemorrhagic, as we can see on this pattern of lung infarction. In some specific cases of necrosis called gangrene, the color of necrotized tissue could be black. Sometimes when the necrotized area is infected by putrefactive microorganisms, some very specific unpleasant odor may appear. In majority cases it happens at wet gangrene. Depending on edematous changes or drying of the organ or drying of the necrotized area, the size of the organ could change. These changes usually are not big. The causes of necrosis are the same of degeneration, and all the causes could be divided into physical, chemical, or biological agents. Based on the causes, the necrosis is divided into 5 types. This is so-called etiological classification of the necrosis. We distinguish traumatic necrosis caused by any physical agent, toxic necrosis caused by any chemical agent, including poisons and some drugs, trophoneuritic necrosis caused by disturbances in tissue innervation that leads to circulation impairment of this area, then allergic necrosis caused by allergic reactions, and vascular necrosis, also called ischemic necrosis, due to decrease of blood supply of tissue. According to mechanism of necrosis development, 2 types are distinguished: direct necrosis and indirect necrosis. In direct necrosis, the pathologic agent influences directly to cell or tissue, causing their death. In indirect necrosis, the pathologic agent leads to appearance of some pathological processes which lead to cell or tissue necrosis. Traumatic and toxic necrosis belong to direct necrosis, whereas in trophoneuritic, allergic, and vascular necrosis, indirect mechanism is present. The main classification of necrosis is clinical morphological classification. According to this classification, the following types of necrosis are distinguished. Coagulation necrosis, also called dry necrosis. This type of necrosis occurs in tissues poor of water and rich of proteins. Types of coagulation necrosis are waxy necrosis, cessation necrosis, fibrinoid necrosis, and fat necrosis. The next type is liquefactive necrosis, also called wet necrosis or colliquation necrosis. This necrosis occurs in tissues poor of proteins and rich of water. Infarction occurs due to decrease of blood supply of the organ or tissue. Death due to ischemia. Gangrene is a specific type of necrosis that occurs in the tissues and parts of the body which are in contact with atmospheric air. Sequestration. It is a type of necrosis when the necrotized tissue does not undergo fusion. Decubitus, also called bedsore, is a kind of gangrene that occurs under long-term pressure of soft tissues between bones and the surface on which patient lies. Waxy necrosis, also called Sanker's necrosis. It occurs mainly at severe infections, in particular under typhoid, and it occurs in the skeletal muscles of thigh or anti-abdominal wall. Under this necrosis, the muscle becomes pale in color, loses its elasticity. In the muscle, we can find some areas of hemorrhages. Under microscopic investigation, the myofibrils look like swollen, increased in size, defragmented, so they are destroyed, and surrounded by inflammatory reaction. Cessation necrosis. It is a kind of necrosis that occurs at a number of infections, in particular tuberculosis, syphilis, and some others. In this necrosis, an area of necrotized tissue separated from surrounding tissue occurs. The necrotic tissue looks like whitish, yellowish or whitish, grayish, dry masses, fragile, and under microscopic investigation they have an appearance of granular eosinophilic masses surrounded by a specific reaction in the form of granuloma. This is a type of coagulation necrosis. The next type of coagulation necrosis is fibrinoid necrosis. This type of necrosis mainly occurs under some autoimmune diseases, generally under immunopathologic reactions or under hypertensive disease in the vessels. Generally, fibrinoid necrosis occurs in the connective tissue, different organs. Under microscopic investigation, the fibrinoid necrosis has a bright eosinophilic appearance. In this particular case, it is in the wall of the vessel. And on this slide, we can see a fibrinoid necrosis area in the glomerulus of the kidney. The fat necrosis, also called steatonecrosis, could be of two types, enzymatic and non-enzymatic, also called traumatic. The enzymatic type occurs in the pancreas. This happens due to activation of pancreatic enzymes which penetrate into the intercellular space of pancreatic gland and destroy the adipocytes normally present in pancreas. On this slide we can see on left side normal adipocytes and on right side the destroyed adipocytes. We see that the presence of light eosinophilic masses, destroyed adipocytes. At this, free acids appear and which bind to calcium salts, and these is presented macroscopically in the form of small-sized whitish dots in the pancreas. Sometimes they unite each other, making bigger area. The non-enzymatic fat necrosis, also called traumatic, mainly occurs in breast fat or subcutaneous or retroperitoneal fat, and is caused by trauma or injury, mechanical injury, under which the adipocytes are destroyed and enzymes are released which further destroy the adipocytes, surrounding adipocytes. The liquefactive or wet necrosis. The usual or typical location for wet necrosis is brain. Under ischemic necrosis or infarction of the brain, this condition is called encephalomalacia, that indicates on softening of the brain tissue at ischemic infarction. The softening of the tissue occurs due to fusion of dead tissue. Here we can see the area, ischemic area, the necrotized tissue, softening of the brain tissue which becomes yellowish, and appearance of cyst in the necrotic area. The cyst formation is an outcome of wet necrosis in brain. As it was said before, the wet necrosis mainly occurs in tissues poor of proteins and rich of fluid. Sometimes the secondary colliquation also may happen. The secondary colliquation term is used for the cases when the primary coagulated necrotic area undergoes fusion and turns to become wet necrosis. This is secondary colliquation. Usually it occurs under some purulent infections and leads to abscess formation. Actually, this is the second type of wet necrosis. Infarction is the next clinical morphological form of necrosis caused by organ or tissue ischemia. The causes of infarction are thrombosis, embolism, or prolonged arterial construction. As you see, all these causes leads to tissue ischemia. We also should mention that besides these causes, the collateral circulation of the tissue or organ, as well as their workload or their functional state, is also very important for infarction formation. We distinguish two types of infarction due to their appearance, due to their shape. According to the shape, the infarction could be conical, as it is in here, in kidney, or irregular shape, as it is in here, for example, in myocardium. It depends on the arterial network pattern. It could be of two types, magistral or network pattern. In case of magistral pattern, which is usually present in lung, kidney, spleen, and some other organs, the conical type of infarction occurs. In brain, intestine, and heart, the irregular shape is formed due to network vascular pattern. According to organ appearance at necrosis, we also distinguish white or ischemic type, red or hemorrhagic type, and the third one, which is white with hemorrhagic rim. This depends on blood supply condition of this organ or tissue. If the organ is rich of collateral circulation, then highly possible hemorrhagic or red infarction occurs, as we can see it here in on this pattern of necrosis of lung tissue. The necrotized area is dense and reddish. The white or ischemic necrosis occurs in the tissues poor of collateral blood supply, as we can see it here. Then the necrotized area is pale, yellowish or yellowish whitish or grayish. Sometimes white with hemorrhagic rim type of infarction occurs mainly in heart and kidney, as we can see it here. This occurs due to initial paralysis of the vessels on the peripheral area of the necrotized tissue, and later these paralyzed vessels are filled with the blood from surrounding collateral arteries, and this leads to small hemorrhages on the peripheral area of the necrotic area. On these slides we can see two patterns of infarction. Here's the kidney infarction. We can see a big area of necrotized renal parenchyma surrounded by inflammatory reaction called demarcation inflammation and hemorrhagic reaction, diabetic hemorrhages with hyperemic vessels. On this slide we can see a hemorrhagic infarction in the lung. We can see presence of blood in the lung parenchyma and destruction of interalveolar septa. Gangrene is a clinical morphological type of necrosis that occurs in the tissues contacting with atmospheric air. It could be of three types: dry gangrene, wet gangrene, and gas gangrene. The dry gangrene usually occurs on the limbs, usually lower limbs. The dry gangrene caused by arterial occlusion, and at this gangrene the tissues become dry, black in color and dense. So mummification process predominates in this case. We also can see a distinctly seen demarcation line. Sometimes the necrotized tissue could be separated from the alive or healthy tissue, and this process is called mutilation. This is typical for dry gangrene only. The wet gangrene can be found in the limbs. However, it is more typical for visceral organs like intestine. In this case, arterial occlusion is followed with venous congestion that makes or creates condition for wet gangrene. Putrefactive infection is also found in wet gangrene. In dry gangrene we find no infection in majority cases. In wet gangrene the tissues are edematous and have grayish brown color, as we can see here on the foot. Also, the demarcation line is either poorly seen or is absent. And due to putrefactive infection, edema, the septicemia is always marked in wet gangrene. A specific type of wet gangrene is noma, which is found usually in kids under severe infections like measles. It occurs on the cheek, also could be found in the perineum area. Under gas gangrene, anaerobic flora is found in the necrotized tissue. In this gangrene, the necrosis is fast, and the anaerobic flora produces a gas which makes a very specific symptom typical for gas gangrene, crepitation. It is a crackling when present on the tissues. The next clinical morphological form of necrosis is sequestration. In this, the necrotized area or necrotized tissue does not undergo autolysis and lies among healthy tissues. In time, the necrotized area is separated from the healthy tissue by connective tissue, making a capsule, sequestration capsule, and inside this capsule a purulent inflammation occurs, leading to purulent cavity appearance. Further, the sequestration can undergo defragmentation, and the purulent process may fuse the surrounding tissue, making a hole or opening this cavity to the environment. This is called fistula, and some fragments of sequester could leave this cavity through the fistula. Bed sore is a kind of gangrene that occurs on the surface of the body, which undergo constant pressure. This usually occurs in severe patients who have to stay in lying position for long term. Here on this picture you can see the typical places where the bed sore could be found. Bed sore is a kind of trophoneuritic necrosis, and usually is presented by such black color area bottom of necrotized tissue surrounded by purulent inflammatory reaction. The outcomes of the necrosis could be divided into favorable and unfavorable groups. The favorable outcomes of the necrosis are restitution and substitution. The best one is restitution, which is complete recovery of the damaged organ or damaged tissue. It is regeneration of specific parenchymatous tissue. The substitution is a kind of outcome when the regeneration is impossible and the damaged tissues are changed by some other tissue. For example, organization when scarring tissue occurs in the area of necrosis. As we can see on this slide, we can see a connective tissue, which is a scar in the myocardium that is formed as a result of myocardial infarction. The next outcome is encapsulation, when the necrotized area is surrounded by connective tissue, making a capsule, separating the necrotized area from the rest of the tissue. The next outcome is petrification, also called calcinosis. When the calcium salts appear in the necrotized area, is a kind of dystrophic calcification. Incrustation is the deposition of crystals of non-calcium salts in the necrosis area. Ossification, when the area of necrosis undergoes ossification. This usually happens after petrification. The necrotized area also may lead to hyalinosis. And in some cases, under wet necrosis, cyst formation may happen, which is typical for brain. Unfavorable outcomes are presented by sequestration or purulent fusion of necrotized tissue. As it was said before, the purulent fusion leads to abscess formation. Apoptosis. It is a programmed cell death in an alive organism. Controversial to necrosis, the apoptosis is a genetically controlled cell death. The apoptosis, as it was said before, occurs in normal functions and under pathological processes in the organism. The functions of apoptosis are programmed cell destruction that happens during embryogenesis, hormone-dependent organ involution. Examples could be atrophy of the ovary during menopause, cell death in proliferating cell populations like epithelium of the intestinal lining, blood, and some others. Elimination of the part of the tumor cells. This happens under spontaneous tumor regression. Immune cell death at cytokine hypersecretion. Apoptosis is also found under pathologic atrophy of hormone-dependent organs, as it is seen, for example, in the uterus at menopause. In pathologic atrophy of parenchymal organs, if their ductal system is obstructed or when their ducts is obstructed, we also can find apoptosis. Under cell death induced by cytotoxic T lymphocytes, it is, for example, in graft rejection, apoptosis could be found. Elimination of the virus-infected cells also happens due to apoptotic mechanism. Example could be Conselmann's bodies in hepatocytes. There are two main mechanisms of apoptosis. They are mitochondrial or death receptor initiated. The mitochondrial mechanism of apoptosis, it happens due to inner or intracellular signals initiating, finally leading to caspase system or caspase proteins stimulating activation, which initiate cell death. The second mechanism is death receptor initiated, caused by some external influence. During apoptosis, a number of morphologic changes may be found. In particular, the volume of the cell at apoptosis decreases, in contrast to necrosis when the cell size is increased. Then under apoptosis, the chromatin condensation and their appearance on the margin of the nucleus, which is called margination, occurs. Later defragmentation, shrinkage of nucleus occurs. The cytoplasm of the cell also undergoes fragmentation, and cell membrane invaginations leads to cytoplasm separation and apoptotic body formation. The apoptotic body is a small part of the cell surrounded by a cell membrane which contains some fragments of cytoplasm and nucleus. Later, the apoptotic bodies are phagocyted by surrounding cells. On this scanning electron microscopy, we can see a apoptotic cell which makes invagination of cell membrane leading to apoptotic body formation. On this electron microscopy, we can see the margination of the nucleus, fragmentation of the nucleus, and apoptotic body formation. There is a number of differences between necrosis and apoptosis processes. First of all, the size of the cell. Under necrosis, due to swelling, the cell increases in size, whereas in apoptosis, cell reduction occurs due to shrinkage of the cell. The necrosis usually occurs in a number of cells, in the group of cells, whereas apoptosis occurs in single cells. After destruction of the cell contents are ingested by macrophages, whereas in apoptosis, the apoptotic bodies are phagocyted by surrounding cells. Due to destruction of the cell in necrosis, it leads to significant inflammation, which is a reaction of surrounding tissue on the cell components present in intercellular space. As no intracellular components occur in intercellular space, in apoptosis, then no inflammatory reaction could be found. The destruction of the cell in necrosis occurs due to loss of membrane integrity. The apoptosis process is not followed by destruction of cell membrane. This is one of the main distinguishing morphologic signs of apoptosis. As a result, in apoptosis, apoptotic bodies are formed, whereas in necrosis, destruction of cell leads to lysis of necrotized cell. In necrosis, the organelles undergo swelling and lysosomal leakage, whereas in apoptosis, the mitochondria release pro-apoptotic proteins, initiating the mechanism of apoptosis. The DNA undergoes random degradation by endonucleases at necrosis, whereas in apoptosis the chromatin condensation and non-random DNA degradation is found.